Y'all have been learning a little bit about different bioinformatics problems throughout these guest lectures, and now I'll talk about some of the research that I do. But for context, so like my name's Niema. I'm a professor at UCSD, but I actually grew up in Chula Vista, so I graduated Bonita Vista High School class of 2011. I went to UCSD for my undergrad, went to UCSD for graduate school, and then am now a professor at UCSD. So I've been in San Diego pretty much my whole life. So yeah, so I'll kind of talk about my research, but then if anyone has any questions about like, I don't know, my path to get to where I am, or like any questions you might have about like your potential kind of future careers, feel free to ask. But I'm gonna go ahead and share my screen. Okay, and then hopefully folks can see it. Cool. So I'm going to talk about so the talk is titled Real-Time Genomic Surveillance of Pathogens Through Bioinformatics Applications. What does that mean? Basically, throughout the COVID pandemic, I helped develop a virus sequencing pipeline that basically tracked COVID throughout San Diego County. You've had talks from some of my collaborators that also worked with me on this project, but I'm going to talk more about like the computer science-y, bioinformatics-y side of how we did that. So here's an overview of today's talk. I'm going to first give an overview of what this term viral molecular epidemiology means. Then I'm going to talk about a type of analysis called consensus sequence generation. And then I'm going to talk about how we actually can analyze a bunch of viral genome sequences that we collect all throughout San Diego. So first, what is viral molecular epidemiology? So I'm assuming folks have probably seen something like this at some point in their biology classes. So you've probably seen an evolutionary tree, which is kind of showing the evolutionary relationships between different species. So at the bottom of the tree, we call those, so first of all these kind of entities on the tree, we call them nodes. And the nodes at the bottom of the tree, we assume are present day species. We call these leaves. The nodes that are at the bottom of the tree, we call them leaves, and we assume that they're present day species. And then usually the nodes that are up in the tree, so in an evolutionary tree, time kind of is moving forward down the tree. So going up back in time, the nodes internally, so we call these internal nodes, represent the ancestors, which we typically assume to be extinct. And then something that you might not have learned in previous biology classes is that the branches of our evolutionary tree actually have some kind of metric along them. So the branches of our evolutionary tree, their lengths represent evolutionary time. So basically, the branch lengths can be some metric that bigger means more evolution has happened. So it could be things like millions of years in the case of these specific species. It could be things like number of generations if we're talking about like human evolution, or it could even be like number of mutations, because more mutations means more evolutionary time has passed. So hopefully folks have seen this type of thing in the context of species, but it turns out you can actually use this exact same framework of thinking for studying viruses of the same species. So instead of the leaves being different species, you can imagine the leaves are different viruses that like maybe I collect from one person and then a different person and a different person. So each of these leaves is like a viral sample I collected from someone. The only distinction is that the branches are going to be much shorter in terms of time. Like here we're talking about maybe days or weeks time scale versus with species, it was like millions of years, but same exact line of thinking. Okay, so how does this tie into an epidemic? Well, epidemics, you kind of lived through a really bad one, so hopefully people have like kind of come to terms with understanding how it works, but how can we kind of think about it from a computer science standpoint? So we can imagine that there's this underlying contact network where this social contact network is basically representing every single social interaction across which the pathogen can transmit. So the nodes in this contact network, and basically you can think of it as like, so we call this a graph, but that just means a network. So it's a network where the nodes represent individuals in my population, and then the edges, these links that connect different nodes, represent risky contacts across which the pathogen could transmit. So for example, if I'm talking about like COVID, maybe this is a very densely connected graph because if you're in the same room as someone, you're susceptible. That's a contact across which the virus could transmit. Versus like if we're talking about a different virus like HIV, HIV is usually transmitted either through like sexual contact or shared injection drug use. So the network structure is going to be very different if we're modeling those different interactions. So basically, the structure of this network kind of depends on what are the social interactions across which the virus could transmit. And then we have what's called a transmission network, which is a subgraph. It's like a sub-network where all the nodes and all the edges existed in the previous contact network. But this is specifically what are the actual transmission events that happen. So you'll notice that here, the links between people are directed. So transmissions go from somebody to somebody. And you'll also notice that the edges, these links have a time associated with them because the transmission event is from some specific person to some specific person at some specific time. So this is the actual transmissions that did happen. And again, depending on the virus and depending on like how fast it transmits and stuff like that, the structure of this and like the times between these transmission events is going to look different, but it kind of like varies virus to virus. Okay, so I introduced evolutionary trees. I introduced transmissions, but how do these two things kind of connect with each other? If we zoom in on like this single transmission event, we can kind of see inside of the person that was initially infected. So again, back in time is up in the tree. Kind of at the time of infection, this person was infected by like one specific viral lineage. And then over time, that one virus kind of evolved in their body. So over time, it evolved, it like branched out, it mutated. And then at present day, at the bottom of the tree, this is like right before the transmission event. So basically, there's all these different viruses at the time of transmission. So in this framework, we can kind of imagine that the transmission is like you're randomly plucking one of these lineages from present day, and then that one lineage that you randomly plucked is going to start a similar process in this recipient. So I'm going to click on the next slide, but I want you to both double check, like take a look at what happens in the recipient, and then also kind of take a look at what happens at the bottom of the host's evolutionary tree as well. So when I click next, we see that a similar evolutionary process is happening in this person, but also time didn't stop for the person who did the transmission. These viruses continue to evolve as time progresses. Okay, so now how do we actually like observe these evolutionary trees? Well, I'm assuming probably everyone here has like done a nasal swab at some point in the past 5 years, probably too many nasal swabs. When you do a nasal swab, you're not like absorbing all the COVID in your body. That would be pretty cool if you could just do a nasal swab and like all the COVID gets sucked up. What you're actually doing is you do the nasal swab and you're just kind of like randomly catching whatever virus particles happen to be in your nasal passage at that time. So you can kind of imagine that like there's all these different viral particles that exist at present day, and you're kind of randomly plucking whatever you happen to grab onto. So this is kind of equivalent to we're observing just these colored dots. There's all these other branches that still exist in the person, but we just don't get to see them because they stayed in the person. So basically, what that means is there's this complete evolutionary history of like every single viral particle that existed in anybody. And then there's kind of this constrained evolutionary tree, which is the best that we could possibly hope to observe, which is basically the leaves of this observed tree are the viral particles that we like randomly grabbed when we did a nasal swab, and then everything up in the tree. So any evolutionary relationships between these that we can hope to infer. This is kind of the best that we could theoretically do. Okay, well, here's a bunch of like trees and stuff, but we know that biology is actually a bunch of like DNA or RNA sequences. So how does that come into play? Because remember, evolution is really just mutations. That's all it means. Evolving is just mutating. So how does the evolution actually look like? Well, you can kind of imagine you have this common ancestor genome sequence. So this is like the genome sequence of the ancestor of all COVID-19 viruses that have ever existed, or like whatever virus that we're looking at. And you can kind of imagine that as you go down this evolutionary tree, so as you go forward in time, you can imagine that evolution is just random mutations happening on each of these different branches. And then these mutations, they're what we call heritable. So when a mutation happens on one of these branches, it gets inherited, it gets passed down all the way down the tree. So like, for example, on this branch, we have this A mutate into a C, but like, for example, on this branch, we have this C in the middle mutate into a T. That C to T mutation gets inherited all the way down the tree. So then we get the actual genome sequences that we collected from all these people. Okay, so just to kind of summarize what we've done, we've basically described in the world of math, we call this a generative process. Basically, we've described this process that this kind of epidemic stuff happens, where you start with a contact network. This is like all the social interactions that the pathogen could transmit, and we can come up with any number of mathematical models to represent how these connections happen. Then you have the transmission network, which is a random process conditioned on the contact network. So it kind of depends on whatever contact network you had. But we can come up with any number of mathematical models where if you gave me a contact network, how does this virus spread across that network? We have the evolutionary tree, phylogeny. Phylogeny is just a fancy word for evolutionary tree, which is some random process of like branching and lengths and stuff like that that's conditioned upon, that depends upon whatever transmission network we happen to have. And then we have the actual sequences that are some random process, these random mutations that happen that's conditioned upon the evolutionary tree that we have. So kind of each of these steps we can imagine is some random process that depends on the step that came right before it. So my lab, we developed a tool called Favites that basically simulates this end-to-end kind of epidemic framework. So then if you want to like study how did COVID begin, or if you want to study like how is HIV spreading, when we had Natasha Martin give a lecture about like mathematical modeling of HIV, this is the type of thing that we can do where we can kind of like simulate realistic scenarios and see, if I do a certain intervention, how is that going to impact my downstream stuff? What's also useful when we come up with these models is that in reality, we don't get to observe these previous steps. In reality, the best that we can hope to observe is you do a nasal swab, I can sequence the virus that's in that nasal swab. So the best that we can actually collect in reality are these sequences. And by coming up with these mathematical models, we can basically do what we call inference. I can work backwards to see, given these sequences, what was the most likely evolutionary history? Given the evolutionary history, what was the most likely transmission network, or at least properties of the transmission network? And then that inference is super helpful for us to do like some type of public health intervention. So to summarize viral molecular epidemiology, it's basically we can use the properties of the evolution of viruses to study a viral epidemic. So we might answer questions like, how did the virus spread across communities or across the world? So this is a screenshot from a tool called NextStrain, which is basically where a lot of COVID sequences were being published as they came out. And each of these samples that we collect, you do a nasal swab, there's a timestamp associated with that. You did this nasal swab on this day at this time. So we can basically track when did these samples get collected across the world, and that can kind of help us see how quickly is the virus spreading in different communities. We can also answer questions like what transmission clusters exist in our population. So in the early days of COVID, and you might be too young to like remember it too thoroughly, but like there was this big question of, there's this new virus. It spreads from just talking to people. What do we shut down? And there was this question of like, do we stop travel? Do we close down restaurants? Do we close down bars? Do we like close down religious places? So this is a figure from a paper that came out pretty early in the COVID pandemic. And each of these circles represents a person. And the network structure, like these circles and their links, was inferred completely without any of this information. So this structure was inferred just using the sequences. So basically from all these different people, they collected sequences and they connected people if their if their COVID sequences were similar. So the structure was inferred completely from the sequences. But then when you overlay information about what interactions that they had, so like you get all these people, you connect them, and then you go and ask them, hey, how do you know this person? Or like, hey, what were you all interacting in? That's how you get these colors on top of this network. And now with these colors, it tells this very informative story of like, if I'm maybe a public health official or like the governor or something like that, and I'm trying to figure out what do I do? Well, if my choices are travel, blocking travel versus like closing down bars or stuff, I can see that most of the transmissions happened at this bar versus travel was not a super big source of transmission. So this would kind of help me inform my public health officials like, oh, okay, maybe I should let people travel, but I should close down bars or like at least make things outdoors or stuff like that. You can also ask questions like, how is the virus mutating across the epidemic? So here's a plot that just kind of is tracking how certain viral lineages in COVID-19 were like growing or shrinking over time. You can ask questions like, what is the molecular mechanism by which the virus invades our cells? So you can actually try to figure out like, how is the virus functionally like binding to the receptors on our cell, and like, what is the mechanism by which it's infecting us? If you can figure this stuff out, then you can come up with therapeutics, because like you can give someone a drug that targets that receptor. And then you can also ask questions like, as the virus is mutating, are any of those mutations impacting the infectiousness of the virus? So here, this is a three-dimensional structure of the spike protein from COVID-19. So spike protein is kind of like how it gets the name coronavirus. There's those little like jutting out things on the surface of the coronavirus. Those are called spike proteins. And the spike protein is kind of like its secret weapon for invading the cell. And here, this is the three-dimensional structure of the spike protein of COVID-19, where we can actually overlay a specific mutation onto the protein. So here, this is saying at position 323 of the protein sequence, like the amino acids that we have, the amino acid P, which I think is like phenylalanine, maybe. I kind of suck that chemistry, but whatever this amino acid is, mutated into this amino acid, which I think is leucine. So we can actually like superimpose that mutation onto the three-dimensional structure. And then we can see, how do we think based on like physics models, how does that impact the structure of the protein? And therefore, how does that impact how well it invades our cells? And then I don't know any of that stuff, but my interest is, as a computer scientist, how do I translate these questions that have very clear public health implications into computational problems that I can write a Python program, just like you all are learning, or some type of program that answers these questions. Okay, so I'm going to pause here. Are there any questions? Feel free to unmute. Kind of making sense so far? Seems like we're all good. Okay, perfect. Okay, so I'm going to talk about the first kind of major problem in viral epidemiology, which is consensus sequence generation. So before I do that, I'm going to start with an analogy. So imagine that you have a bunch of stacks, you like have a bunch of copies of the exact same newspaper. So you have a stack of this exact newspaper where all of these copies are identical. And now imagine that you're a crazy person and you put a bunch of dynamite under those newspapers. I'm a professional. Don't try this at home. Imagine you light the fuse. And then poof, these newspapers kind of blow up. And all you're left with are these like tiny charred fragments of newspaper. And your question is, how can I reconstruct what that newspaper said from all these tiny burnt up pieces of newspaper? So maybe I'll pause. Does anyone have any ideas? How might we like try to reconstruct this newspaper from these little pieces? If I like forced you to try to do it as like a 4th of July activity, I'd blow up a newspaper until you reconstruct it. What would you do?
I guess the best you could do is just try to find like pieces that are the least like damaged and try to put them
Mhm.
together.
And then what would you do with them? So like, let's say I find a couple pieces that are like not super damaged, just kind of like a little tiny, but not damaged. What would you do with them?
I would put them side by side to try to recognize where they belong, to try to make
Exactly.
sense of it.
Perfect. Absolutely. You'd probably be like, well, wait up. This part of this newspaper looks similar to this one. Let me try to put them side by side and see, oh, I can kind of like overlay them. And now I can reconstruct the longer piece. And you kind of keep putting these overlaps on top of each other to try to rebuild what that newspaper said. So it turns out that this like crazy problem that I've introduced of blowing up all these newspapers is very similar to a problem in bioinformatics known as genome assembly. So there's this virus that has some unknown genome. The way that we actually try to sequence the genome of that virus is we create a bunch of copies of that genome. There's a technique known as PCR. I don't know if folks have learned about PCR in biology class. So yeah, oh, perfect. Okay. Yeah, polymerase chain reaction. You can kind of imagine it's like a copy machine for DNA sequences, where basically we take the genome and it just replicates, 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 replicates. we get all these copies of the same exact genome. And then for reasons you don't have to worry about, the sequencing machines that we use, they're not able to just like read the genome from start to end. They can only read relatively small fragments. So for example, the COVID genome is like 30,000 letters long, but the DNA sequencing machines that we have can only sequence like 100 to 150 letters long. So what we actually do in the sequencing experiment is we randomly chop up all these copies. So we break them down into all these like random smaller pieces. And then the DNA sequencing machine kind of, you can imagine you're like closing your eyes and just like randomly grabbing whatever fragments that you can. So the sequencing machine randomly sequences some of those. And our goal is from those tiny fragments, how do we look at their overlaps to kind of reconstruct the whole genome sequence? And before I go on, as a fun piece of trivia, has anyone heard about the company Illumina? Has that come up in biology classes a little bit? Maybe not. No, it's all good. So Illumina, it's the world leader in DNA sequencing technologies. Like every single biologist in the world uses Illumina sequencing. And it's actually in San Diego. So if you ever take the 805 North, it's like just west of the freeway, or I think it's on the 805. When you get to like La Jolla Village Drive, close to UCSD, it's right off the freeway. It's like 5 minutes from UCSD. So if you're in San Diego, you actually are in the most important city in the world for DNA sequencing, which is pretty crazy. Just a fun piece of trivia. So before I move on, I also want to introduce the concept of a reference genome. So has the term reference genome come up in like a biology class? Like you might have heard about, well, like the human reference genome? Maybe not. Kind of. Awesome. So I'll just define it anyway. So a reference genome, you can imagine it's a high confidence assembled genome sequence that is constructed as a representative example of an individual organism. And what do I mean by representative example? Well, everybody on this call, I'm assuming you're all human. If you're not, that's okay. You're still welcome here. But if everyone here is human, we all have almost identical genome sequences. It's like 99.9% identical. We know that there's definitely a little bit of difference, but they're almost identical. So a reference genome, it's like a genome sequence that we construct, which every single entity in that species is going to be a little bit different from that reference genome, but they're going to be almost identical. It's going to be like super, super similar. So historically, some kind of like brief history over here, the human genome was assembled in 2001. That was actually before y'all were born. Huh, that's crazy. Dang, I'm old. So I was like a little kid when this happened. But yeah, the human genome sequence was assembled in 2001. And that was like this big multi-billion-dollar effort between like the government and a bunch of companies. And then over time, it's become easier and easier to assemble new genome sequences. So like the mouse genome was assembled next, then the rat genome. Why might you think mouse and rat were the next two that got assembled? Any thoughts? Just out of like curiosity. Not just because they're cute. Any ideas? It's all good. If not, that's okay. So basically, a lot of like drug testing and stuff like that, or a lot of like scientific experiments we do on mouse models because they're similar to humans, but like we don't, you can't like do experimentation on actual humans. So like mice and rats, they're like very common for experiments that you do before you do human testing. And same with chimpanzee. So chimpanzee is very closely related to humans. So, these genomes are helpful because a lot of biomedical research you do testing on these types of animals before you do it on humans. But yeah, so as you can see, like over time, we faster and faster were able to assemble new genomes. And like what used to be something that like only the entire US government with like a bunch of money could do, now you can actually do pretty easily in your own lab. Like instead of it taking many years and a bunch of money, now it's like maybe on the order of like hundreds of thousands of dollars or like a couple $1,000,000, which like sounds like a lot, but is not super huge in the grand scheme of things. And like individual research labs now are able to do this kind of routinely just because DNA sequencing is much cheaper and the algorithms to assemble these genomes are a lot faster. However, it still usually takes on the order of like maybe a couple years. With the COVID pandemic, though, we had kind of a breakthrough in research where very, very, very early on in the pandemic, the reference genome sequence of the COVID-19, like the SARS-CoV-2 virus, was published. And I just want to kind of highlight this paper was published in February 3rd, 2020. So paper publications, usually you have to like do the research, then you get the results, then you write the paper, then you submit the paper, it goes through review, and it's this like lengthy process. So like just getting a paper published usually takes like a month or so after you finish the research. So November 2019, like late November 2019, early December 2019, that's when like anyone even caught wind that something was going on. And this paper was published February 3rd, 2020. So this is like a month after the pandemic started. We had a high confidence reference genome. Also, fun bit of trivia. This guy's one of my collaborators, so I know this guy. Um, but yeah, so very early on in the pandemic, we had a reference genome at our disposal. So what this helps us do is if you have a reference genome, it makes it much easier to reconstruct the genome sequences of different samples you collect. So like if you did a nasal swab and I want to sequence what does the virus look like in here, I don't have to do that like randomly blowing up the genome and then like trying to reconstruct it from scratch thing. I can use that reference genome to make my problem a lot easier. So here, imagine that the blue fragments are from this longer blue genome that's the one that came from your sample, and this green genome is the reference genome. So we know for a fact that the blue genome sequence that I'm trying to reconstruct differs a little bit from the reference genome, just like how your genome differs from my genome a little bit, but they're almost identical. They're super, 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 super similar. So now instead of trying to find overlaps between the genomes, like these small fragments themselves, I could compare each of these fragments against my reference genome to figure out where they most line up. And now reconstructing the genome sequence becomes super easy. I can just kind of go line by line by line and see what's the most frequent letter at any of these positions in the reference genome as they appear in my blue fragments. Let me output that as the next letter of my genome sequence. So short answer is, with this reference genome, this problem becomes much computationally easier and also much cheaper biologically. So the tool to do this, some people published a tool in January 2019, so very timely, that implements this exact thing. Also, fun fact, this guy, Christian Anderson, this guy is one of my collaborators as well. He was actually like on the original calls for like when COVID was first breaking out, like Anthony Fauci had like emailed him and a couple other people to like try to figure out what the hell's going on. So big person in this field. Um, but with COVID-19 pandemic, viral genome sequencing became super cheap. So we actually had an explosion in how much data we were collecting, that even this tool wasn't fast enough. So in the early days of the COVID pandemic, I was brought in to try to like speed things up a bit. So I took their tool and I kind of figured out how can I re-implement the same idea, but much faster. So over here, this is a plot comparing the runtime of their tool, shown in red, versus the runtime of my tool, shown in green, for doing this genome sequence reconstruction. The horizontal axis represents the number of reads, so the number of those small fragments. So right on the horizontal axis means bigger data set. And the vertical axis shows the runtime in terms of seconds. And you can see that it's like, I don't know, like 500 times faster or something like that. So like when their tool takes maybe like a few minutes or like even an hour or so, my tool runs in like a second or like 2 seconds. So super fast. And then in terms of memory, so like your computer has RAM, so like you might have like a computer that has like 4 GB of RAM or like 8 GB of RAM. That memory is basically the memory that your programs have that they can use to store whatever they're using kind of while they're running. So if you write a bioinformatics tool that uses too much memory, I don't know if you've ever opened like too many tabs on Chrome and then your computer starts freaking out. But basically, the more memory you use, the worse it is, because you need more expensive computers that have enough memory to run your analysis. So here the red line is showing their tool's memory usage, and the green line is showing my tool's memory usage. So here you'll notice that like on this biggest data point, their tool takes like 1 GB of memory, and as you get bigger, it takes even more and more memory, versus like my tool is consistently right around like 10 megabytes or so, so pretty small. And then in terms of accuracy, the vertical axis here is showing, so I basically simulated a bunch of data, like, I simulated a bunch of sequencing data from genomes that we already knew, and then I tried to see how well do each of these two pipelines reconstruct them. So vertical axis here shows error, and then horizontal axis is showing a bunch of different COVID lineages sequenced at different sequencing amounts. So this 30x versus 40x versus 50x, you don't have to worry about what that means, but just bigger number means we generated more sequencing data. And you'll notice that across the board, my tool is like just as accurate as the other tool. So that's a good kind of sanity check. If you want to run this yourself, you can actually run it using this tool called ViralWasm. This is a tool that one of the students in my lab developed. And you can run it just on your phone, on your laptop, anywhere that you want. And you can actually like analyze real genome sequence data from different viruses just completely in your browser without having to install any tools. Okay, so I'm going to pause here again. Any questions about what I just talked about? Feel free to type in the chat or feel free to unmute. So short answer is basically now we have a way of, if we have a bunch of people doing nasal swabs or something like that, I can figure out what is the genome sequence in this nasal swab super, super fast and super cheaply. Okay, so what does that mean? That means that a lot of people are going to do this. So during the COVID pandemic, and this screenshot might actually be out of date. Like this is how many genome sequences were collected all throughout the world. This is 20 million genome sequences. That's a lot of genome sequences. And my hot take is this is actually too small. So way more than 20 million people got COVID. And from my perspective, because genome sequencing is getting so cheap and because these tools are so fast, we should sequence everybody. So in the future, this number is going to get even bigger. And in this part of the talk, I'm going to talk about like, okay, now I've collected all these millions of genome sequences. What do I actually do with them? So the standard workflow that we typically do is we start with these unaligned genome sequences. These are genome sequences that we just collect from all these different people, and we use the pipeline I just talked about, like that tool that I just talked about a second ago, to reconstruct all their genome sequences, but they're not aligned. So what does unaligned mean? It means that like there could be deletions. So for example, maybe like these two sequences have C's over here, but it looks like that's a deletion in these sequences. You might have learned about like insertions in your biology classes. And then when we're doing that genome reconstruction, sometimes we miss segments of the genome just because like maybe we didn't sequence it carefully enough. So there's all sorts of missing parts of the genome sequences, whether it's actual deletions or we just missed part of the genome, that the first step is just figuring out how do I get these genomes to line up more nicely. So basically figuring out where do these little spaces go so that I can get these genomes to line up more carefully, like more nicely. So has anyone heard of the term homology? Like homologous genes, homologous phenotypes. Yeah. So do you remember like what does homology mean if you had to like put in your best words? No pressure, but if you want.
It's oh, I forgot. But the opposite of like the homologous is heterozygous, and it has to do with, no,
Oh,
wait.
you're thinking homozygous.
Oh yeah, huh.
Yeah, yeah. So,
No,
so that's good. So
wait.
homozygous, so this is like humans have two copies of every chromosome. So homozygous means we have the same exact allele on both copies. Heterozygous means that we have different. So same exact root. So homo, that root means same. So homologous, you can think of as shared, like shared features that were both inherited from the same common ancestor. So for example, we look a lot like monkeys. So like we have 5 fingers on each hand. We have 5 toes on each foot. We even have a tailbone, even though we no longer have tails, but we still have a tailbone. These are properties that were inherited from the same common ancestor. So homologous means a feature that two different things have that was inherited from the same exact feature in their common ancestor. So we usually think about it, and you actually even see this with like bats. So the wings of a bat are actually homologous to human hands. It's like basically the hands of some common ancestor that then got this like webbing between them and like got all weird shaped and stuff. So we think about homology usually in the context of like features, like physical features, but you can actually think about homology at the genome sequence level as well, where like when I line these genomes up nicely, each column represents the nucleotide that was inherited from the same exact position in the common ancestor. So even though these different sequences might have different letters, like this one has a C in this first position, it corresponds to whatever letter was inherited from the common ancestor of all of them that just mutated into a C at some point in this specific one. Or maybe some letter from the common ancestor that got deleted in these ones. So when we put these gaps in between them to get them to line up, not only does it look nicer, but it's actually, we're figuring out sequence level homology, which is going to be the key piece of information that helps us infer their evolutionary history. It turns out that this computational problem, even though I've described it super simply, is very, 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 very hard. It's very slow. So tools exist that try to implement this, but these tools were not built to scale to like millions of genome sequences. These were meant for like hundreds or like maybe thousands. So when the COVID-19 pandemic hit, we needed stuff that was much faster. So these tools, though, what they're trying to do is they're trying to align completely arbitrary sequences. So you could take this tool mapped and you can throw any random sequences you want at it, and it's going to try to line them up. But our problem is actually much easier than that. Our problem is not how do we get a bunch of arbitrary sequences to line up. Our problem is we have millions of sequences that are almost identical. And also we have a reference genome that all of them are almost identical too. So instead of trying to figure out how to line up arbitrary sequences, we're actually trying to line up sequences that are almost identical. So I came up with an approach that basically, instead of trying to get these, so here the green genome sequence is the reference genome, and all these other colors are like the individual samples we've collected from people. Instead of trying to line them up with each other, let me independently do an alignment with the reference genome. And then once I get those alignments, I can basically use my reference genome to reconstruct the multiple sequence line. I can basically see for the first position of my reference genome, what letter in each of these sequences lined up to that first letter of my reference genome. Let me just output all those letters as the first column of my reference genome. Do the same thing with the second column, with the third column, with the fourth column, etc., until I reconstruct the whole reference genome. So this is a technique that I implemented in a tool called Viral MSA. And over here, I'm showing the runtime of my tool versus the current best practice tools. So the horizontal axis represents how many genome sequences we have. This was SARS-CoV-2. So yeah, how many genome sequences we're analyzing. The vertical axis is the runtime in seconds. And then this red tool, that was the one that I showed you called MAFT, which was like prior to my tool, it was the best practice tool that people used. And then this green line is another tool that implements the idea that I had of align to reference, but I don't know why it's super slow, but I just included it just to show like, I don't know, maybe to flex, who knows. But yeah, basically you can see that, that was a joke. But you can see that my tool is like way faster than any of these tools by like 1,000x or more. And actually, when we get to like 40,000 genome sequences, it only takes, I don't know, like 100 seconds. That's like, you know, a couple minutes. So much more scalable. In addition to that, my student also implemented a web application that you're actually going to be using next week as part of your action items. You're going to be running a real SARS-CoV-2 kind of evolutionary history inference thing using this tool. But basically, yeah, you can run my tool as well as a bunch of other stuff directly in the web browser. Okay, so the next step is phylogenetic inference. So given this multiple sequence alignment, we want to be able to infer what is the most likely evolutionary tree. And the trees that we get, for reasons that you don't have to worry about, is that we basically are unable to just directly infer the directionality of evolution. So we have no way of knowing, like, did an A mutate into a T, or did a T mutate into an A? We don't know directionality, just like how many mutations happened. So because of that, we don't know the directionality of evolution, which is forward in time. So we don't know where the root of this evolutionary tree is. We know the general structure of it, and we know the distances, but we don't know where is the ancestor so that we would be able to figure out the evolutionary history, like forward in time. But still, this is super helpful. So phylogenetic inference, basically, given that multiple sequence alignment, we want to infer what is the most likely evolutionary tree that came up with these sequences. Basically, the way that we do this is we come up with models of sequence evolution to define how likely different tree structures are, and then we try to find the tree that's best. This problem, you don't have to worry about what this NP hard word means, but it's basically super, super challenging. It takes a lot of time to run perfectly. So people come up with algorithms that are not going to give you the optimal tree, but hopefully are faster and give you a good enough tree. I'm just going to kind of quickly click through these. You don't have to worry about these just because we're running out of time. Um, but basically, there's a bunch of different approaches that exist that use different statistics and probability kind of techniques to try to infer the best evolutionary tree. And don't worry about this, but later on, if you ever take like a statistics course, some of these terms are going to be a little bit more relevant. Um, Even these heuristics, they're kind of slow, but some of them seem kind of promising. But don't worry about this. But basically, we got a bunch of tools that try to infer the evolutionary tree. And then there's also all these downstream things like how do I find where the root of this evolutionary tree is? If I can find the root of this tree, that tells me where is the common ancestor, and it tells me the directionality of evolution. And then from there, there's all sorts of downstream analyses like how do I infer the ancestral states? Like how do I infer what were the ancestral genome sequences and therefore like where all the mutations happened? How do I like transmission cluster? So I showed you that graph earlier, like that network of all the different people that like socialize at a bar versus like at a traveling or wedding or whatever. So how do I cluster people into transmission clusters based on the evolutionary tree or like based on the sequences? All sorts of downstream things that we want to do that I'm just going to kind of click through these for the sake of time. But basically, a lot of tools that do that. So I'm going to talk a little bit about how do we actually do that clustering. So again, the goal of this clustering is, let's say I've come up with all these genome sequences, I've aligned them, I maybe have the evolutionary tree. How do I group people together? And the whole goal of this molecular clustering, this like transmission clustering, is I want to know which people are most likely connected together in the transmission network. If I can figure that out, that helps me inform my public health policy. So for example, if I notice that like there's a big growing population of people that are contracting HIV, I want to be able to group them together to figure out like what is the shared kind of like mode of transmission between these people. Because if you imagine like if I see that it's sexual contact, that tells me, oh, well, maybe I should have some type of intervention of like maybe giving people free contraceptives, or there's a treatment called PrEP. It stands for pre-exposure prophylaxis, that basically someone who doesn't have HIV but has partners who do have HIV, you can take this treatment that basically is kind of like a shield that protects you from HIV. There's also antiretroviral therapy that like if your partner has HIV, someone living with HIV could take antiretroviral therapy, ART, we call it, and that basically suppresses the virus in their body to like prevent transmission. Versus if you're talking about like, what if it was because of shared injection drug use? Maybe HIV is blowing up and you find out that like the growing clusters are because of shared injection drug use. That tells you maybe I should have like clean needle exchange programs. Maybe I should invest in like harm reduction. Maybe I should have like other types of information that I'm disseminating about like drug use and stuff like that. So very different interventions depending on how the transmissions are happening, and clustering helps us figure that out. So first I'm going to talk about how we can do clustering from sequences. So imagine there's two people that have my virus. I'm going to call them X and Y, and I collect a viral genome sequence from person X and a viral genome sequence from person Y. If X and Y were both infected from the same person, not a lot of evolution has happened between their genome sequences. So the pairwise distance between the genome sequences that I collect, just like the number of mutations between these two genome sequences, is going to be super, 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 super tiny. If X and Y were not infected from the same person, but they were still infected from the same kind of general group of transmissions, their pairwise distance, the number of mutations between their viral genomes, is going to be pretty small. Not as small as the first case, but still pretty small. And then if X and Y were infected from completely unrelated sources, So like when we talked about with Natasha Martin when she gave a talk a couple of weeks ago, or like last week or something, she mentioned like the COVID surveillance that we were doing on UCSD campus. Well, maybe two people got infected from each other, but maybe, okay, well, this is a college campus. People are moving in from all over the world, even though they're both infected with COVID on campus, maybe they're from completely unrelated sources wherever they came from. So if this is the scenario, a lot of mutations have happened between their two genome sequences. So the pairwise distance, the number of mutations, is going to be pretty large. So this kind of pattern, it helps motivate a fairly simple idea where, well, I can basically take two people and I can link them together if their pairwise distance between their viral genomes, so the number of mutations between their viral genomes, is small enough. So what I could do is for every pair of individuals, let me calculate their number of mutations between their viruses, and then let me like link them together if it's small. And then I can output each resulting chain of links as a molecular cluster. So here's a small example. Imagine I have four people, A, B, C, and D, and these dashed lines represent their pairwise distances. So like A and B has a pairwise distance of 0.01. So this means that like 1% of their genomes is mutated with respect to each other. So like 1% difference. A to C maybe is 2% different. Maybe B to D is 5% different. And let's imagine that I'm using a pairwise distance threshold of 1.5%. So I actually do this for the CDC. I build tools for calculating these molecular networks for HIV surveillance at the CDC. And for the CDC HIV surveillance, they use 1.5%. So let's use that in this example. What I would do is basically see out of all these pairwise distances, which one is less than 1.5%. Let me link those pairs of individuals. And now let me output those groups that I've connected as my transmission cluster. So I would say A, B, and C are closely related, but D is kind of separate from them. And this is a tool that's implemented in this HIV trace tool that's largely used for HIV epidemiology, but we've also used it for COVID epidemiology. And you might recognize this name from the very first guest lecture. So Joel is one of my colleagues that he was from the very first guest lecture in this program. And then also my student implemented it into this web app that you're going to be using next week. So that's how you can do clustering from sequences. So I have a bunch of sequences. I'm just directly comparing pairs of sequences. What if I have an evolutionary tree? So as you saw earlier, the evolutionary tree is very heavily constrained by the transmission network. So if X and Y are infected from the same person, if I have this evolutionary tree of all the genome sequences, X and Y are going to be very close together on that evolutionary tree. So this like global evolutionary tree, they're going to be like next to each other. Versus if X and Y were not infected from the same person, but they're still kind of closely related in the same transmission group, they're not going to be like right next to each other on the evolutionary tree, but they're going to be, still pretty close together. So like if I have the whole global COVID-19 evolutionary tree, these two people are going to be like in the same general subtree, not right next to each other, but pretty close together. Versus if X and Y were infected from completely unrelated sources, they're going to be very far apart in the evolutionary tree. So like if I have the global evolutionary tree, they're going to be in like completely arbitrary parts of the tree. So similarly, I can define clusters using relationships in this evolutionary tree. And there's a lot of ways that I could define clusters. I could maybe say, I want to cut the tree into subtrees that have a maximum pairwise distance below some threshold. Or maybe I cut super long branches or all sorts of stuff. And I could also do that exact same type of clustering that I did with the sequences, but instead of defining their pairwise distances using like how many mutations there exist between these two sequences, I could define pairwise distances as like distances between two leaves on my tree. So like the path between two things I could treat as the distance, all sorts of stuff. So back in 2019, me and some colleagues implemented a tool called Tree Cluster that basically does this. You give it some definition of clustering, you give it an evolutionary tree, and it clusters the sequences using that evolutionary tree. And the beauty of this is that it's super, 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 super fast. So here I'm comparing its performance against HIV trace and this other evolutionary tree clustering tool called Cluster Picker. And again, horizontal axis is data set size, vertical axis is runtime. My tool is super, super, super fast. How do I enable this? There's other tools that I've built, like TreeSwift is a tool that you can basically, in Python, it's a Python package that you can use to like analyze evolutionary trees. So when we get to the project phase in this summer program, this might be something that you're going to potentially use in your research projects. I recently built another tool that's also a package for doing stuff with evolutionary trees. This one's in C++. Um, Same general idea. Horizontal axis is data set size, vertical axis. And this one is how much memory is the tool using, and this one is runtime for just like loading a tree. And these kind of get repetitive, but basically my tools are, whoops, these three. So this one, and I think it's the other one's also behind it, maybe. Yeah. And then same over here. Like these ones and this one are my tools. So my tools are like, again, much faster and much lower memory. Okay, so overall summary, basically my interests as a computer scientist are we have big biological data. A lot of it is very medically relevant. And how do we translate a lot of these like public health or biological questions into computer science problems? And then my research is how do I come up with the fastest or most efficient solutions, like the most efficient Python programs or C++ programs or whatever that solve those problems so that then I can give the public health officials or epidemiologists really fast tools to fight this public health crisis. So, yeah, thanks for listening to me ramble. Any questions about anything, either about the research or just anything that you're curious about? Feel free to unmute, but no pressure. Yeah,
Um,
Marty, go for
I
it.
I wanted to ask, I think you already like went over it, but I was gonna ask like what kind of data or are you like collecting from the rats and how is it like processed or analyzed using computer science techniques?
Yeah, for sure. Collected from the what? I didn't catch that last word.
Um, Collecting from the rats. I heard that you were
Oh,
talking
from the
about how
rats.
rats and like mice are similar to our um DNA and all.
Oh, that's a
How are
great
you
question.
collecting that?
Yeah. So let me show you. So here's a database called GenBank. So GenBank, it's this big database of a bunch of genome sequencing data. I'm going to post the link in the chat. And basically you can search for whatever um, type of data set that you're looking for. So let's imagine that I want like, I think the mouse Latin name is musculus. Is that what it was? I'm forgetting. I could probably just also search for the name of the rat, but yeah, so I can like search for whatever species I'm looking at, and then like they have this, cute picture of a rat. Or this is a mouse. This is not a rat. Mice and rats are different, but this is a mouse. And I can actually like look up like let me look at its genome sequence. So I can click on see genome data viewer, and this is loading, and like it's actually like all the chromosomes of the mouse genome, and I can actually like click on a specific part and can I like zoom in? Oh, too many genes in that region. There's probably like a zoom button somewhere. Um, yeah, either way. But yeah, so it basically this genome annotation, it's showing like what are the different genes. So here this is like a gene called XKR4, whatever that means. Something related to blood group. And I can actually like click on these. And then does it let me go in? There we go. So now I've actually like zoomed in on a specific gene, and like in genetics classes, you might not have learned about, I guess have they taught you about like transcription? Is that something that's been covered yet?
No,
No, it's all good.
not
So
true.
transcription is basically so your DNA of an organism has all the information for all the genes. So it has the actual DNA sequence of every single gene in your in your organism. And then transcription is how you go from DNA to RNA. So you basically when you want to use a gene, like when the cell wants to use a gene, it transcribes the gene. It basically converts a segment of that DNA into RNA. And then that RNA is then translated into protein. And then proteins are kind of like the functional unit of the cell that actually do stuff. So here, these arrows it's actually showing, because DNA is double-stranded, so some genes are on one strand, some genes are on the opposite strand. So these arrows are actually showing what direction on the genome transcription is happening. And yeah, can I zoom in further? Let me see if I can, I don't know how far in it lets me zoom in, but eventually, can I like do a small segment? Zoom on range. Ooh, zoom to sequence. That's what I want to do. Cool. And now I can actually like look at the letter by letter by letter by letter genome sequence of the mouse genome. Um, you can also compare genomes. So you can like compare the mouse genome versus like the rat genome. Let's see if they have that. Or actually, let's do mouse against human, because that was what your question started with. Like, why do we even study mice? I can actually compare their genomes and then see, and this looks messy, but basically what this plot is showing is this top one is the mouse genome with all the chromosomes kind of lined up next to each other. And this bottom one is the human genome with all the chromosomes lined up to each other. And these little ribbons are showing, I talked about homology earlier. Homologous parts of your genome are parts of your genome that were inherited from the same common ancestors. So, for example, this part of chromosome 3 on mouse corresponds to like this part of chromosome 1 in human. And those were inherited from the same exact part of the genome of some common ancestor between mice and humans. So this way, like, let's imagine I'm trying to design some drug that, I don't know, is like treating some specific gene or something like that. Like, let's say there's some human gene that causes some disease if it's broken, and I want to come up with a drug that helps target that specific gene. Well, I can't just test it in humans directly, but like, let's say that gene is like right here on the human genome. I could see, this is getting kind of hard to follow. Let's see, does it go? Maybe it's over here. I kind of like lost the ribbon, but I can basically see like what gene in mice is the corresponding homologous gene. And I can now do research into that to kind of like study it in mice and then try to use those results to then try to figure out like, how do I study it in humans safely? All sorts of stuff. So yeah, definitely play around with this website. You can also like find raw sequencing data. So I can go to, let's see, all nucleotide sequences. So you can like find all the genes and like I can click on any of these and then it shows me like the actual DNA sequence of this specific protein. And it also shows me the protein sequence. So this is an amino acid sequence. And is this a, let's see, is this a protein receptor? Yeah, protein receptor domain containing protein. Yeah, okay. Yeah, hopefully that kind of answers your question. Sorry, I kind of rambled for a little bit.
No, it did. Thank you.
Yeah,
I
no
have
problem.
one more question.
Yeah, go for it.
Um, what inspired you to pursue computer science, and what current research projects are you doing right now?
Yeah, great question. So computers in general, I was like always super interested when I was in high school and like middle school. But like in terms of my like professional interest, like I always was kind of interested in pursuing a career in medicine. So like I grew up like I always wanted to be a doctor. And then like when I was in high school, I applied to college to like a biology major, trying to go like the pre-med route to like hopefully become a doctor one day. But in terms of my hobbies, I always really liked computers. And I thought that these two fields were separate from each other. I didn't know that like you could use computers to study biomedical research. And then when I came to UCSD, so UCSD, I had no idea that like it's actually the top university for bioinformatics in the world. It has like the #1 bioinformatics program. I just stayed in San Diego because I grew up in San Diego and all my buddies stayed at like most of my friends went to SDSU, so I was like, oh, I want to go to UCSD, so I'm like pretty close by. Um, I didn't really know that it was like a world leader in bioinformatics. So when I got into college, I started like going to research talks, just to kind of learn more about what bioinformatics was. And then I switched majors, and that's where I learned that oh, hey, I can actually use computers to study biomedical research And then through research that like as an undergrad, I was doing research projects under the supervision of different professors. And like that got me even more excited because I like the kind of I don't know, in high school I really liked math classes a lot. Like I took calculus and like I really enjoyed all my math classes. Um, and I feel like computer science is like puzzle solving in a sense, where like you have this specific problem and you're trying to build a tool that then solves that problem. And you're kind of seeing this in the Python book that you're working through, where like we have all these interesting problems that we're trying to solve. So to me, it kind of feels like solving a puzzle, but then when you solve it, the results are helping people with their health. So that's kind of what got me excited about it. Yeah, I don't know if that kind of answers the
No,
question.
that does. Thank you.
Yeah, yeah, no problem. Yeah, any other questions? I have plenty of time, so feel free.
No, those were all my questions. Actually,
Yeah.
I noticed that you said you went in with biology, and that's like the major I was looking into
Nice.
going in. So when you went into that major, um, how am I gonna fix this question? Was it like I don't know how to ask this question.
Take your time, no rush.
Um, because I wanted to do biology, but I'm also
Mhm.
taking a AP biology class, but I'm
Oh,
like
nice.
trying to see because I'm also trying to go somewhere into like medical, but dentistry
Hmm.
actually. Um, so I'm trying to see if it's like kind of corresponding. Like, what made you what was the major what you were expecting? What I'm trying to say. Like
Like
in
what?
that
Sorry, I cut
major,
off a little bit. Sorry, go for it.
sorry.
No, you're
In
good.
that major, like, was it what you were expecting? Was it somewhat similar to what you were learning in high school, or like completely different from what you were
Yeah, yeah,
expecting?
good question. So, yeah, that's a great question. First of all, you're already ahead of the game because I didn't take AP Bio in high school and I kind of like regretted it when I got into college because I was doing a lot of biology classes, but like I feel like biology as a field, there's a lot of terminology. I mean, like as we saw in this talk, things like homology, things like homozygous versus heterozygous, things like phylogeny. Like, what do any of these words mean? Like, it's the whole language that you have to kind of learn. And I feel like the more repetitive exposure that you get to it, you start kind of picking up the key words over time. So I kind of wish I had taken AP Bio, and I took AP Physics because I just like math a lot. Ended up not actually pursuing physics at all, but I just like math. But I feel like if I had taken AP Bio, that would have given me a little bit more early exposure to like some of these concepts that then when you start college, so your intro classes are almost identical to your AP classes. So like if you take AP Bio, then your first year of bio classes in college are like almost identical. You might actually even test out of them. When I took AP Calc, I took AP Calc BC. I don't know. I think Benita still might have it. I don't know if we had to like I had to take geometry in the summer at some point to like get a little bit ahead to be able to take Calc BC. But AP Calc BC, I think I tested out of the first two college calculus classes, and then even the third class, a lot of it overlapped with the end of Calc BC, and then there was like a couple weeks of new stuff. So each of the AP classes that you take in high school, I think they either directly map to like the first intro class that you would take, but you might actually test out of, or even if you don't get to test out of something, like even if they still make you take it, it probably overlaps with like a good half of the content in that corresponding college intro class. But then like the rest of your college is building on that. So I think kind of each stage of your academics, you're kind of like in high school, you're learning the foundations of these specific topics. And you actually, if you go a little bit further back, like when you were in middle school, you probably remember like science class. It wasn't even like biology or chemistry or physics or whatever. It was like science. And you're learning the foundations of like all of science. And then you got into high school and now you're like specializing and learning the more kind of advanced topics in each of these fields. When you get into college, it's even more of that, where like you take even more advanced biology that builds on what you did in AP Bio, even more advanced math than you did in like maybe calculus or statistics or whatever, even more advanced chemistry, whatever. And then when you get into, if you ever pursue graduate school, even further. So like graduate school classes, like in a master's or like medical school or something like that, they build even further. So it's going to look similar, but kind of going deeper in, if that kind of makes sense. Like you're kind of dipping your toes in the water right now, and then like you keep going deeper and deeper into the subject as you get further. Yeah, great question.
Okay, thank you.
Yeah, yeah, no problem. And I realized I forgot to, you had also asked like what type of research do I do? So I'm just gonna post this link in the chat. I
Mhm.
have like a list of all the research projects I've been working on. You can kind of look through them if you're interested. But the overarching summary of my research is like basically I have collaborators that are like at the School of Medicine or in the biological sciences or whatever. And they have different analyses that they want to do that are too slow. And they usually just kind of like tap me in of like, I'm the guy that makes slow things run faster. So I just kind of get tapped in, make their stuff run fast, and then like we publish a paper about it. Um, yeah, and anything that you've learned about like COVID origins, like if you've heard anything about like raccoon dog stuff, like oh there was a wet market where like we think that COVID jumped from a raccoon dog or something into humans. That was all me and Joel that you learned about from week one. Um, Pretty weird stuff.
I remember when you were talking about just like you said when they bring you in to like make it go faster. When you were trying to like I remember the computer part where you made your notice how like the data and like the memory was like going up for it. It took a
Mhm,
longer time, but for years it stayed consistent.
mhm, mhm.
With you doing that, how long did it take you for you to make it have a consistent data for the memory?
Yeah,
Stay
yeah. So
consistent?
like how long does it run, or like how long did it take me to like figure it out?
How long did it take you to figure out how to
Oh, that's a
keep the
good question.
consistency?
Yeah, yeah. So I mean, and this is something that I really like about computer science is like once you so when you're first learning Python, like we call it a programming language because it really is a language. Like you're learning a brand new language, just like how if you learn Spanish or like Mandarin or some language, like you're just learning how to communicate with your computer when you're learning the basics. And then once you get comfortable with that, in the same way that like when you first learned how to like speak English, it was a lot of like grammar and syntax like back in elementary school and like spelling. And then now that you're in high school, you're doing things like you already know how to spell, you know how to write sentences. So now you can like write essays where you're like you know the syntax like the back of your hand, and now you can do more creative, like higher level thinking, and you're using English as your vehicle for like writing these more beautiful thoughts of like stories or essays or whatever. So computer science is very much the same way. We're like Python, what you're doing right now is kind of like when you were learning spelling and grammar and stuff in elementary school. And then once you get the hang of how to code in Python and you're not like stumbling over the syntax and stuff, which you will get to with more and more practice, then the fun really begins. Where like you're like basically solving problems, and Python is kind of your vehicle to translate those problems to your computer. So, um, how long did it take me? It's kind of funny. So like when I solve these problems, I don't know if it's like something wrong with my brain or something like that, but like when one of these problems comes up, my brain just like fixates on it. And like actually most of these solutions I kind of come up with either in the shower or like sometimes when I'm about to go to sleep, I'm just kind of like not really dreaming, but I'm just kind of like thinking about it and then like visualizing how do I think it's gonna look like. And then once I figure it out, like in the shower or like when I'm about to sleep or like just going on walks and stuff, I have an idea and I'm like, oh crap, let me try that. And then I like try to implement it and then see how it works. So for that specific one, I think actually like writing it only took me maybe like a day or so, but that was in the same way that like when you write a story you don't just like write the story, you first do all this brainstorming in advance. So I think the thinking about it took like months. I was just kind of like listening to how people were doing it, thinking about like, oh, how would I do it differently, all that type of stuff. And then once it actually comes to implementing, it was much faster. And let me also real quick, let me paste this link in the chat. So that was one specific tool, but the first thing that kind of got me into all this viral sequencing stuff in the first place was like in the early days of the COVID pandemic, basically, so there's this Illumina sequencing machine called a NovaSeq. You don't have to worry about what that means, but I could actually probably maybe pull it up. So there's an Illumina sequencing machine that this is like some big old machine that you like put the raw DNA. Oh, what the heck? Why is it so tiny? Yeah, why are these pictures so tiny? I want a bigger picture. There we go. Is this big? Okay, nice. So yeah, so this is their most bleeding edge, like recent sequencing machine, and you basically open up a compartment of it, put your DNA sample, and then it runs this experiment where it like does PCR and it does all the stuff that you need to actually get the reads, like the actual sequence data. So one DNA sequencing run on this machine sequences like a terabyte roughly of data. So like you've dealt with like gigabytes, megabytes, stuff like that. So 1 terabyte of data, so 1,000 gigabytes gets produced every time you run one of these. And the actual like biological experiment that it's running takes roughly 24 hours. So in the early days of the COVID pandemic, the San Diego County of Public Health hit up one of my collaborators, Rob Knight. He's like a big person in the gut microbiome world, like everything related to bacteria. He's like one of the big people in that space. And then they basically tapped him in as like, hey, we want to be able to sequence COVID all over San Diego, both in wastewater. So like if you've pooped at any point during the COVID pandemic in San Diego, we have sequenced your poop at some point. Just a fun fact. But yeah, like basically they wanted to do like mass sequencing of wastewater and also mass sequencing of COVID tests that were positive to be able to track COVID in the county. So they were collecting enough samples to basically run one of these experiments every single day. And it takes 24 hours to churn out a terabyte of data. But then the data analysis using the tools that I showed you from that guy, Christian Anderson, takes like 2 weeks of compute time to analyze 1 terabyte of data. So this was like a big problem because if we're generating data every 24 hours, we need to analyze it in at most 24 hours to stay up to date. Otherwise, we fall behind immediately. So then I was kind of tapped in of like, hey, this is taking 2 weeks. We need it less than 24 hours. How do we fix it? So this link that I just pasted in the chat, this is like the results of, we had just phone calls pretty much every day with like different experts where I was just trying to learn like what are they doing right now? What are some ideas that I have for speeding it up? So these were like the quick and dirty notes that I took, just kind of keeping track of like what are these tools, and like you'll even see like some of the notes that I left myself, like I wonder if this thing is fine, or like to speed up this thing I could do this. Even here, like there's notes like, this is a flag that Karthik was like one of my colleagues. This is a flag Karthik told me to add. I don't know what it does, but let me add it. Like why does this thing need a sort of blah, blah, blah. Like I kind of left myself notes on what I could do. So this, like actually solving everything, I think was the result of maybe like 3 or 4 months of just meetings every day to like, freaking out. We were all like, what do we do? What do we do? What do we do? And then once I kind of took my notes, understood the problem, and like started building it, it only took maybe like a couple of days to actually write. So a lot of thinking and then a little bit of writing.
Cool.
Yeah, thank you. Yeah, great questions. Awesome.
Thank you
Alaska,
again for
oh yeah,
answering.
sorry.
Thank you again for answering my questions.
Yeah, absolutely. And Alaska, did you have any questions? No pressure, but just wanted to check.
Um, not today. Everything was pretty clear. It was great. Thank you.
Perfect. Yeah, no problem. Okay, cool. Well, yeah, thanks so much for listening to me ramble about bioinformatics for 45 minutes or hour 15 minutes. Never mind, sorry. And by the way, like if either of you are ever interested in like biology department at UCSD or like learning more about the different majors or stuff like that, like feel free to let me know and I can connect you with professors that I know in the different departments. So, definitely leverage the connections you are making in this program. I'm happy to help you kind of meet up with people and ask them questions. Awesome. Cool. Yeah, thank you for listening to me and have a great 4th of July. I will see you all later. Bye, everybody.
Thank you. Bye.